So hi everyone. So welcome to the webinar hosted by the Scientific Committee. Just for the housekeeping about the Histiocyte Society. So it's a non-profit group of more than 200 physicians and scientists from around the globe committed to advancing knowledge and improving outcomes for patients with histiocytic disorders, with a focus on clinical and basic research and education. And this session is proudly hosted by the Scientific Committee. So for the housekeeping, every participants are muted, and you should submit your comments via the chat feature on your webinar panel, and the chat will be seen by our staff and panelists. The chat will be in the controls of the bottom of your screen, and after clicking, the chat window will appear on the right. Then you can type your message and press enter to send it. And if then you can put the questions in the Q&A box, and they will be addressed at the end. So please indicate who the question is for, and please put questions for staff in the chat box, and you can like the questions in the Q&A box if your questions has already been asked by someone else. And due to time constraints, we will not be able to take all the questions. The webinar is being recorded and will be sent to all the registrants, and the Histiocyte Society members will also be able to find the video in the members section of the website. And we thank our sponsor, which is the Histiocytosis Association and the Cincinnati Children's. So Dina is going to start.
Hi, everyone. So I'm Deanna Fournier, Executive Director of the Histiocytosis Association, and I'm going to kick today's session off with a little bit of background about the association, as well as how the Histiocyte Society and the association partner together to work on our annual grants program. So just really quickly, the important work of the association, if you aren't aware of the ways that we connect with the histio community and the support that we provide. We focus on research, trials, registries, and other studies related to all histiocytic disorders, provide emotional support to patients' families through outreach, events, and virtual opportunities, provide educational resources on all histiocytic disorders. We have a physician's directory, and we ensure support along every step of their journey with histiocytosis. And then we seek to share results and key learning and patient experiences with the community to expand knowledge and awareness. So all of the work that we do rolls into those areas. We've partnered with the Histiocyte Society for nearly 35 years, and the association has served as secretariat to the society to provide the administrative support needed to optimize focus on research. And that partnership is demonstrated every year, most significantly through the annual grant funding process of the Histiocytosis Association. The funding decisions are made by the association's board of trustees, and they're based upon the reviews and evaluation of the competitive grant applications by the Histiocyte Society's scientific committee. And that provides the basis for that review provides the basis for all of our decisions. And so it's incredibly important for us that that relationship exists and that we work together. There are a lot of different elements of the grant process, and I will be talking a lot about that today. The grant process supports scientific research investigating the causes, mechanisms, and improved means of treatment of histiocytic disorders. RFRPs are evaluated based on science, feasibility, and relevance to histiocytosis. And all the proposals are expected to address one or more of the histiocytic disorders. So that's really the background of what we're looking for. Some years I know we have looked at specific priorities, but we are always open to accepting all grant applications as long as they are based in relevance to histiocytosis and meet the requirements. And then the grant applications face an extensive evaluation and peer review process. And we've modeled that by the model that the NIH has. It's a US National Institutes of health. And the reason why we selected that process years ago was it ensures an impartial and transparent approach, and it makes sure that it's fair, equitable, timely, and free of bias. And so that's another reason why the association does not initially evaluate and review the grants, but we are very appreciative of the support of the Histiocyte Society's scientific committee to help us with those reviews. And then ultimately we take the recommendations and make a final decision through our board of trustees. Just to give a picture of what that relationship looks like. So the Histiocytosis Association, our team has 7 full-time employees that support all areas of our mission. Our board of trustees currently is made up of 9 volunteer members, and we have various committees. We have our strategic committee, our fundraising committee, our finance committee, our governance committee, and then our scientific committee. And our scientific committee on our board advises the association's board and the association team on anything, priorities, initiatives that we have that fall into that scientific umbrella. The Histiocyte Society, as you know, is a separate nonprofit organization, over 250 members, and there's a separate board of trustees. And the scientific committee of the Histiocyte Society is helpful and responsible for reviewing applications for our annual grant process. So just so you can see how that relationship exists, the scientific committee will take those applications, review them, and then pass recommendations back to the scientific committee of our board, who then has that discussion with our board of trustees to make a final decision. So hopefully that was helpful background, and I'm happy to answer more questions later, but I'm going to turn it back to Julian to take you through the rest of the presentation.
Thank you, Dina. So I'll take you through the grading process. And so as part of the ongoing partnership between the Histiocyte Society and the Histiocytosis Association, the scientific committee of the society scores the research grant proposals of the association's research grant program each year. So the preparation for grading, so the Histiocytosis Association manages the grading portal and process. All scoring happens in a secure grading portal that is similar to the application portal, and the Society's scientific committee submits conflict of interest for each application at the start of grading to ensure a fair and unbiased grading process. So it's, as Dina told you, designed similarly to the US NIH grading process. So you see here that there is an overall impact with it's graded like 1, 2, 3 for high, and mainly high is high importance interest in the field, and sometimes there are some or no weaknesses. 4, 5, 6 is medium. It's usually high or medium or moderate application with some or major weaknesses, and low is usually when you have moderate or high importance in the field but many weaknesses, or low or no importance with some or no weaknesses. So for instance, you can have a 5, which is a good medium impact application, and the entire scale should always be considered. So here are the 4 grading criteria. You have the scientific impact, feasibility, relevance to histiocytic disorders, and overall impact, and each gets a 1 to 9, 1 being exceptional. Then all graders provide numerical scores on all applications unless there is a conflict of interest. Each application is assigned a primary, secondary, and tertiary grader to leave written comments, and these are assigned based on disease expertise by the scientific committee chair. And after scores are submitted, the scientific committee chair determines if any additional reviews is needed. If none, scores are prepared for the scientific committee discussion, which is held during the Histiocyte Society annual meeting. Then the applications and scoring is discussed by the scientific committee and representative of the HR scientific committee. After the discussion, each member of the society's scientific committee submits a final 1 to 9 score recommendation on each application through the secure portal. The society's scientific committee chair submits a report and recommendation for funding to the Histiocytosis Association Scientific Committee for consideration. And at the end, the final selection of the grant award is made by the HA. And when making the final determination, they consider all applications, the priorities and goal of the association's research grant program, the recommendation of its scientific committee, the grading and recommendation of the Histiocytosis committee, scientific committee, and the HA's available funding. So each year there is a certain number of grants depending on from year to year. It's not always the same number of grants which are given. So after that, Astrid will make a presentation. So I leave you with Astrid for the first presentation.
Yeah, okay. Can you hear me? Can you hear me now? Yep, okay. So on behalf of the core project team depicted here on the right, which consists of Oussama Abla, Cor van den Bos, Jan van Laar on the right, myself, and the PhD student involved, Paul Kemps, I give you a short overview of the project that we submitted at the Histiocytosis Association in 2014. So the basis of our grant application was really the landmark publication by Barrett Rollins group showing the presence of recurrent BRAF mutations in the Langerhans cell histiocytosis. And obviously, when Barrett published these data, a lot of questions came around about what's the clinical impact of this particular somatic mutation. So Oussama, Cor, and myself sat down on one of the Histiocyte Society meetings, and we discussed to submit a proposal to the Histiocytosis Association to really investigate this in more detail. And this was partly driven by an observation made in the same time that Barrett was working on the molecular analysis by my PhD student, Willemijn Kwispel, who started to really dig out the immune infiltrates that you see in classical histiocytosis lesions. And what you can see here is immunohistochemistry of T cells stained in red using the CD3 antibody, at the activation marker CD25 depicted in blue, and FOXP3 in green, staining the nuclei in green, showing that the cells that express high levels of CD25 and FOXP3 positive nucleus, these are the Tregs. But obviously in these LCH lesions, you also find activated T cells expressing high levels of CD25 without the FOXP3 expression. So these seem like bona fide conventional T cells that have been activated in situ. So the question, of course, is if histiocytes express this mutant BRAF protein, is this protein actually intracellularly processed into small peptides, and do these mutant peptides or neoepitopes actually appear at the cell surface? What is depicted here? So at the cell surface, all HLA class 1 molecules express peptides that have been intracellularly processed. And if this comes from a mutant protein, then you would expect epitopes to be expressed at the cell surface, and this may trigger the T cells present in the lesion. So this is the proposal that we submitted in 2014. So with me and Cor van den Bos being the PIs from the Netherlands and Oussama, the PI from Canada. This was the basic setup. We would request the patients to give a buccal swab. So we use that to isolate germline DNA for HLA genotyping. We collected the biopsies of these patients to do the molecular analysis, case report forms, and also assess the degree of lesional infiltration by CD3 positive, CD8 positive T cells. Here you can see all the institutes that joined in. So it's the LUMC, Amsterdam Medical Center, Erasmus Medical Center, and Sick Kids Hospital in Toronto. So if you're interested, the question is actually, or the answer to the question whether these peptides appear at the cell surface is actually no. If you're interested, please read this paper. So this is the first paper that came out of the results that we collected from this pilot grant supported by the Histiocyte Society. And when we were working on getting all these genotype data available, we were actually scooped by the French National Histiocytosis Research Group, who were the first to publish the clinical impact of the BRAF mutation on higher prevalence of reactivations in patients expressing this mutation as compared to the patients who did not express the BRAF V600E mutations depicted here in yellow. Obviously, that BRAF wild-type group contained a lot of patients expressing any of the other identified MAP kinase activating mutations. So we were, of course, a bit disappointed that we were not the first to report our data, but we decided not to cry for too long and really work on understanding what is the impact of the other mutations that may be expressed by these BRAF wild-type patients. So we submitted a small grant proposal to a very small private foundation in the Netherlands called 1000 Kaartjes voor Jultje. And I did it together with Tom van Wezel, who is the head of the molecular diagnostics of the pathology department in my institute. And they gave us 14,000 euros to actually extend the diagnostic cancer hotspot panel that the team of Tom is using for the molecular diagnostics. And we quite easily successfully extended the panel. And then we applied for a follow-up grant with Histio UK. And in this grant, we've actually proposed to do cost-efficient genetic profiling on patients enrolled in LCH4. Because I talked with Milne again at one of the Histiocyte Society meetings, and Milne agreed with me that it would really be a wasted opportunity not to do the genotype analysis in conjunction to the international LCH4 study. So in 2019, we presented the interim results of our analysis. We then had genotype and clinical data available of 179 patients, and we already showed very nicely that indeed the type of BRAF mutation matters, because if you have an exon 12 mutation, the patients actually perform better than if you have the BRAF V600E mutation or any of the other mutations that we specifically addressed. And this, I think, is a very nice picture of Paul showing that he's very proud to receive the Nesbitt Prize in Clinical Science. So at this point, we are closing the study finally. So 5 years of data collection. And we now have in total 366 pediatric genotype patients. About half of the cohort consists of LCH4 patients. The other cohort is a retrospective cohort from the Dutch and the Canadian patients. And among the 190 LCH4 genotype patients, we have patients from 5 different countries: Denmark, Czech Republic, Austria, Italy, and the Netherlands. So the take-home message, I think, of the story of my grant is that if you can organize seed funding, for instance, via the Histiocytosis Association, and if you can then manage to generate some pilot data, which are really calling for more money, you can, so this grant that we received was actually also supported by the 1000 Kaartjes voor Jultje, the Belgian foundation called Kiwanis Run for LCH, and the Histio UK. So all together, we received 100,000 funding altogether, and there's a technician and a PhD student that worked for a few years on this grant, and together with a very active community from the European Consortium for Histiocytosis, I think this was really the solution to get so many genotype patients. So I hope my talk can inspire you, and I really would like to encourage you to submit a grant, because it can really these kind of projects can really fly off once you have your seed capital. Thank you.
Thank you very much, Astrid. And now we'll move to Egle Kvedaraite, who is in Karolinska in Stockholm and who did some of her work in Singapore. So Egle.
Yeah, thank you. Can you hear me okay?
Yeah, perfect.
Yeah, so unfortunately, Florent could not make it today, but we are really thankful for the grant that allowed us to do this work that I'm going to talk about. So basically what I will tell you today is our kind of major finding is, and it is that LCH cells seem to arise then in fact from both dendritic cell and monocyte DC3 lineages. And I will not go through the details of LCH because you're experts on that, but I would like to give you some details on the aims that we had when we started off. So basically what we wanted to do is to identify the core signature and then heterogeneity of lesional tumor cells using single cell analysis, because it's the first key step to separate the neoplastic cells from the normal MMPs or mononuclear phagocytes in the lesions, to then be able to look at the heterogeneity within. And then we wanted to validate this, these markers and pathways also on protein level. And so how did we do this? We took aspirates from lesional samples, and then we, for each cell, got 18, like a panel of 18 markers on flow cytometry, and combined this with single cell RNA-seq. So we had a transcriptome of each cell, which was sequenced quite deep, up to 1 million reads. And then we integrated this to tell exactly which cell is which for every cell in the lesional microenvironment. And then after that, we tried to validate this, both using another data set that we kindly used, Dr. Hatter kindly provided. And then we also validated this on protein level. And the second validation step was to, of course, look in the microenvironment and see if we can find these populations there as well. By the way, can you see my pointer? Yes? Okay, so this was the aims and study design. And then what did we find? So first of all, we were able to delineate LCH cells from the rest of the mononuclear phagocytes on protein level, as shown here, but also, and importantly, on RNA level. And what is even more important than that, those identities overlapped. So now we could see that, these are LCH cells. So by the way, for the color coding, when you see a pink cell through the presentation, it is an LCH cells, and then LCH cell, and then mononuclear phagocytes are depicted in other colors, like PDC is in green, and then DC is in this color, and monocytes in blue, and then MREG DC is in this orange, light orange. Okay, so we did that, and then we looked that it looked good in all the patients, and then after that, we of course looked at the genes so that we accurately, could identify each cell. And after that, so yeah, I mean, we are immunologists, many of us, so this question is perhaps obvious, but I still wanted to give an example to why it is so important as like first delineation step, because for example, we see one cluster 1, that is positive for CCR7, LAMP3, CD83, and, LCH cells express very, very low levels of those molecules, if any. And in fact, we have here so only last year, Miriam and I published this paper saying that, in fact, in tumorous lesions, we have those MREG DCs that we believe are identified by this cluster, and we also find it in LCH lesions. But imagine if we could not delineate those cells from the rest of the LCH cells, then it would have appeared as a subcluster or something like that. So this is just to illustrate why this is important. And so after that, we of course looked at how these cells looked in terms of their expression pattern compared to so we compared LCH cells with the rest of the phagocytes in the lesions. And then we saw very strong signatures pointing to senescence, both on gene level and then pathway level. And this has been touched upon already by the group of Miriam and that you most of you have heard about previously. But But we could also then see that in the patient lesions and not in an animal model. And then, okay, so we did that. And then we looked specifically in LCH cells. And when we did exactly the same pipeline, but only including LCH cells. And then we found 2 clusters that were still, they were not like completely separated. So there was some convergence in those, but they were distinct. Also, again, on all those levels. And then, so we summarized that, in fact, we perhaps have 2 major clusters. And summary this far is that, yeah, as I told you, LCH cells could be separated from normal counterparts. And then senescence was a very strong signature in it. And then 2 major clusters. And the next question was, okay, look at those 2 major clusters. How are they related to the normal immune system, normal MMPs that are also found at the site? So this is just a brief refreshing of the MMP system. MMP system is mononuclear phagocytes. We have DC3s, we have DC1, 2s, etc., etc. So to answer this question, we then took the signatures of those cells and then mapped the LCH cells. We asked what is the degree of similarity between every single LCH cell and those signatures. And then we found that, in fact, if you put monocyte / DC3, so in fact, we took here DC3 signature, but DC3 signature is very, very similar to monocytes. So then we put this signature in the up here, and then DC2 signature below, and then we asked every cell to, end up with one of those signatures. And then we could clearly see that, in fact, so cluster 0, separation here, and cluster 1, separation here. And then when we mapped them back in our single cell map, we could see that, so if you can recognize this is one cluster, it mainly enriches mono DC3 signature, and then DC2 cells, they ended up in this other cluster. So this was super exciting. And then we said, okay, how do we validate this? So for this, we took another data set that, as I mentioned, was kindly provided by Dr. Hutter. And then we integrated those 2 data sets because they also had very many cells. They analyzed the cells using 10x. And this is the main difference from our work where we used SmartSeq2. And then we can see that we have a nice integration of all those cells. We have clusters 1 and cluster 0. And then importantly, so all the gray cells are 10x, and the ones that are depicted are our cells. And then when we map them back in this map, I don't know if we can again appreciate this converging pattern, right? That we have. So the cells that ended up with monocyte DC3s, they are green in here, they end up in this branch. And then you have DC2-like cells that end up in the upper branch like this. And then they just mix up together like that. So it's not like clear separation between the 2, as you can appreciate here from this image. Okay, I have the slide just to remind us what is the difference between 10. I mean, there are so many different techniques, and I'm not going to go through all of them, but it's good to have a little bit of background. So this is quality control of each single cell. And then you see that, so the red one here is SmartSeq2, and then the rest is every other sample from the 10x data set that is already published. And then you can see that, so in terms of numbers of genes, it's not there is a little bit of difference, but not so big of a difference. And the same thing with mitochondrial genes, that is also another quality control detail. But the major difference is the depth. Here we go to 1 million, and here we get just a couple of thousand, which means that basically the resolution differs highly. And that's why we need both methods, because we get complementary information from the both. Okay, and then so this was really exciting to check how the developmental trajectory would look. So if you remember, we got DC2-like cells here, and then we thought, how do they develop? And here, based on RNA velocity, and we used very, very many different methods, but I show you one because we don't have very much time. But here, for example, you can appreciate that all the arrows point from this cluster in, so it feeds from here to this big blob. And then you can see, so using stochastic model, you can see small arrows coming here that become even more convincing when you use dynamic modeling of RNA velocity. So RNA velocity is a method where you use spliced and unspliced variants of of the gene to tell what is the vector of the time trajectory, because it takes time to go from splice to unsplice, from unsplice to splice. Anyway, so it kind of confirms our hypothesis that in fact it comes like this from two directions and diverge into some kind of thing that you cannot really different, like it's not completely, not like T cells and B cells or whatever, but it's really, so they come like this and then converge. And of course, very importantly, we found the BRAF in those what we depicted LCH cells, but not in the non-LCH cells. And this is very important. We did this using ddPCR and having heterogeneous and homo cell lines for the BRAF 600E to check that our controls are okay. And really all the LCH cells are like, as we know, they have one wild type and one BRAF 600E allele. So this is the summary, that we could see the polarization towards DCs and monocytes / DC3s to major developmental trajectories, and then that they carry the mutation. And how did they look in the different tissue environments? That's another question. And to validate this, we did this high-dimensional imaging called Maxima, where you basically take an antibody and put a PE on top of it. And then what you do is that you remove PE, and then you can have a lot of antibodies. Like we here used 30 antibodies to image this and looked outside and inside of the lesion. And here is how skin looks. So basically, you can appreciate cytokeratin and then LCH cells here. And then you can again see these two populations that when we backgate again to the tissue, they seem to be meaningful units that are in fact situated a little bit together across the tissues. And then we looked at very different tissues, but I won't show you every of them. So to sum up, we have senescent Langerhans cells that arise from those two lineages, and that we validated this using protein analysis, cross data set integration, and high dimensional microscopy. And of course, the question that we are mostly excited about is how can we all together use this knowledge to change, like to maybe find new ways of how to target LCH cells, because that's kind of the idea we have. So hopefully everyone could participate in this. And to finish up, we would like to thank everyone, of course, Florent's group and Yaninge's group and all the other groups, Jim Yao, and all the patients and their families who participated very kindly, all the doctors who provided the samples, and nurses, and Histiocytosis Society for the funding, and all other funders listed here. And thank you for listening.
Thank you very much, Egle. So we don't have time for questions, but we'll move on to Paul Mind's presentation from Newcastle.
Okay, yes, so thanks as usual for the invitation to speak here on our grants. So first of all, looking at our 2014 grant that was written by Matthew, this was funded part of my PhD. And from this grant, we managed to get 2 papers. The first was looking at the types of circulating cells which can be induced to bear a Langerhans cell phenotype. And in this brief report, we saw that the CD1C positive DCs could be induced to appear like Langerhans cells with GM-CSF and TGF-beta or EMP7. And then in a longer paper, we looked at the cell types from histiocytosis patients which carry the V600E mutation. We saw that this mutation was present in circulating monocytes and dendritic cells and in the earlier progenitor cells of the bone marrow. So with these 2 papers, I successfully defended my thesis against Carl Allen and managed to graduate. But unfortunately, at no point during my PhD did I manage to grow facial hair as good as Julian's. So looking further forward at the grant from 2019, written by myself, so obviously my interest in histiocytosis has been maintained, and the background of this grant is looking at how hyperactivation of the MAP kinase pathway actually leads to the LCH or ECD cells. As we know, this hyperactivation is the molecular cause of LCH or ECD. We don't know this next step of how this hyperactivation leads to the regulation of transcription of myeloid cells. We know that MAP kinase inhibitors are highly effective in preventing disease in patients with refractory LCH. However, we're not really sure why the V600 alleles persist at such a high level within the peripheral blood. We know that once these inhibitors are removed, there's a rapid relapse of disease. So this failure of the inhibitors to induce molecular remission is currently one of the most pressing clinical problems. So a more detailed understanding of the consequences of hyperactivation and how this is affected by inhibitor therapy. This may identify new targets for intervention capable of synergizing with the MAPK inhibitors and achieve a full molecular remission. So the aim is to determine the effects of ERK hyperactivation on myeloid cell transcription. And to do this, we're looking at TargetSeq, which allows for parallel genotyping and transcriptomic analysis of single cells isolated from the patient's PBMC or bone marrow at diagnosis and also at various stages through their treatment. We want to see which transcriptomic programs are activated, such as it might be possible to see the senescence programs activated, which have been recently described. So the process of TargetSeq is just taking the tumor biopsy, or in this case, the liquid biopsy, which is the PBMC, and sorting the cell subtypes of interest with flow cytometry into single cell plates, going through a number of PCR reactions to then split and get a full single cell genotyping of single cells and a whole transcriptome from those cells as well. So the progress to date, we've recruited several patients. This is an example of a patient with several PBMC samples over time alongside their treatments. So pre-treatment samples, samples on inhibitor, and also samples when inhibitor was removed and rapid relapse occurred. To get the cells of interest, we've got the full sorting panels already in place. We won't go through this in detail, but at the first stage, we'll be looking at the CD14 positive monocytes from the blood, also possibly the dendritic cell subsets. And from the bone marrow, we'll be looking at the CD34 positive progenitor cells with index sorting data so that we know exactly which progenitor cell subset has been sorted. The next step was to design primers for the V600E mutation, and this is fairly difficult because you need to design primers for the cDNA and also the gDNA that are capable of working together. So we've done this and validated now that these primers that we've designed will not adversely interact together. And then the next step was to optimize the actual TargetSeq reaction. So this is using single cells now from the cell line which is positive for V600E, which is displayed in in blue, and another cell line which is positive for a TET2 mutation, highlighted in green. In orange. This is color coded by genotype on this Tisney plot, and you can see that the transcriptional variance between these single cells is easy to pick up. We have a couple of papers out now showing that we can analyze these kind of data from single cells from the progenitor and mature cell subsets. So we are now ready to go. And we also have some really nice liquid handling robots set up. So we're very happy to get on with these experiments now, and we're hoping to be on time for our extended deadline of December this year. So I'm just going to thank everyone in the Human Dendritic Cell Lab, and also in Adam Mead's lab in Oxford, who are collaborating in this research, the core facilities in Newcastle, and the clinicians from around the UK and the LCH team for the samples, as well as elephants research.
All right. Thank you very much, Paul. And then we'll move to Gang Huang from Cincinnati's Children's Hospital in Cincinnati.
Okay, first thank the scientific committee give us the opportunity to present this work. This work and also early work is really supported by the Histiocytosis Association. Without that, the funding support is really impossible. So today I'm focused on the cytokine storm. L10 cooperate with the L18 drive HLH-like disease. So although we are really new to the field, kind of new, but we have some our thinking about the HLH. I think probably everyone familiar with the fire triangle. You need a fuel, oxygen, and heat to guide the fire. And the HLH patient, no matter familial type or secondary HLH, the patient are also on fire. But we believe they also have some predisposition, like mutations, and also some triggers, underlying disease of the infections going to lead to the cytokine storm. And also 5 years ago, we got the findings like support the underlying mechanism. We think probably the HIF-1 alpha glycolysis pathway is really important underlying the primary and secondary HLH, because as you know, T cell and the macrophage activation all rely on the glycolysis HIF-1 alpha pathways. So we have made the transgenic mice of HIF-1 alpha gain function mice. That mice can develop in the B6 background, develop HLH-like. So we published that paper in the 2017. And we know the upstream of HIF-1 alpha are not just oxygen metabolite, but also some cytokine signals can activate HIF-1 alpha. So we started working on the cytokine storm a few years ago. And luckily, last year we got the Histiocytosis grant on this kind of project. So as you know, HLH always associated with the underlying cytokine storm. And from the patient, we know there are, every patient probably is slightly different, but there are a few kind of major elevated cytokines like interferon gamma, L18, and L10. And some of them are really implicated in the activation of the macrophage and the T cells. And but interestingly, in the L10 is always a controversial, because from the immunology field, people always believe TGF-beta L10 is really anti-inflammatory. So we don't really know whether the L10 play a protective role or cooperative role to lead to the HLH. So we really want to dig into this to understand what's the kind of important. As you know, now the people we in the field already target interferon gamma and also the L18. So this is copied from nice review from Alex, and from the HLH, even some maybe the LCH, the genetic predisposition and background inflammatory activity seems really important for disease, and then the triggers, infection, bacteria, fungus, and maybe even some patient with the virus like EBV-CMV. Those kind of triggers eventually lead to cytokine storm, T cell and macrophage activations eventually lead to phenotype. So what's really the trigger contributed to that? So when we look at the profile of the infections, we really think probably L10 is one of the trigger. As you may know, many bacteria, fungus, and they lead to L10 overexpression. And also even some herpes virus like EBV-CMV, they themselves encode the virus type of L10. They can also stimulate the endogenous L10. We think probably the L10, instead of immune suppression, maybe have some cooperative role with L18 or even other cytokines and lead to these conditions. As you know, Dr. Tang is also in the audience. So they published back to 13 years ago, high L10 levels was really an unfavorable prognosis factor for HLH. So we generated the transgenic mice inducible L18, L10 overexpression. So basically it's lock-stop cassette with the 2A, and we can generate the, with deletion of the stop cassette, then we can express the pre-L18 and L10, then they can further process it to the L18. So we can generate a pretty nice high level of L18 and L10 in the mice, and it's very close to the secondary HLH patient level, and the L18 a little bit low range of the patient level. But these mice nonetheless developed a very nice HLH-like phenotype, pancytopenia, splenomegaly, and rapid HLH-like disease progression in this double cytokine transgenic mice. Around 2 months, all the mice die of this HLH phenotype. We look into the spleen and bone marrow, we can find many this kind of phagocytosis type of macrophage, and they can eat 1 cell, 2 cells, even more than kind of 5, 6 cells. And also we measure the ferritin and triglyceride, and also the NK cell activity. You can see they increase you have a ferritin hypertriglyceride. And also we measure the NK cell functions. They have dramatically reduced the CD107 and NK cell activities in the kidney acids. So if you look at the diagnosis criteria, the mouse really have the 6 out of 8 of the HLH. As you know, the mouse HLH, usually they don't have fever. Instead, they have low body temperatures, and we didn't really measure the soluble CD25. So it looks like a HLH. And the disease take almost 2 months, and we think probably one of the reasons is L18 level was not high enough because the range is really low range of the patient L18 level. We know the pro L18 need the inflammasome to process to the mature L18. In some patients, secondary HL patient, they have mutation in the inflammasome pathways to elevate the L1, L18. So we in collaborate with Dr. Rebecca Mars, and we breed our transgenic mice with XAP knockout, which kind of elevated the L18 procession. So now we can see we can have almost 10-fold increase the L18 level in the serum, and also can few-fold further increase the L10. Now you see this is the double transgenic. This with the XAP knockout further increase cytokine level. You see much kind of quicker, around 1 month, all my style of the HLH. Not only that, when now we look at the spleen or bone marrow, we can see even more dramatic phagocytosis, and this different color encode they eat 1 cell, 2 cell, or more than 2 cells. So you see there are many macrophage in the spleen can eat many different blood cells. So because of time, I just kind of briefly introduce the mouse also developed a very severe phenotype. So HSC depletion and also myeloid bias in the bone marrow. So you can see the dual transgenic and the transgenic with XP knockout. They deplete the long-term HSC, dramatically increase the MPP and the GMP. Basically, this is really a acute infection type of phenomenon. It leads to myeloid biased hematopoiesis, and also they dramatically increase the neutrophil and the monocyte in the peripheral and also in some tissue. And also they have active macrophage, have hemophagocytosis. And more importantly, they also have lymphopenia. Both T cell and B cell was significantly reduced. So all this phenomenon really, probably you can recall that's really similar as the COVID. So last year when the COVID phenotype came out, so we really think, oh, this is something like really related to this cytokine storm, myeloid bias, the immune kind of response. And I hope I convince you the L10, instead of protective roles, really cooperate with the L18, can lead to the secondary HLH mass in phenotype in the mouse. And more importantly, whether this cytokine can initiate, whether still important for the therapy. So we did some preliminary data. So we used the LCMV Armstrong and virus and perfect knockout model. We did the therapy with the either block the L10, L18, or use the control interferon gamma as control. So you can see L18, we don't really see a protective role, but blocking L10 or interferon gamma, we can see a significant kind of benefit. We think probably the IL-18 not only contribute the disease, maybe a new therapeutic target for the disease. So this is my summary slide. So I think the cytokine storm really lead to the HLH. There are many ways. Different patient group probably have a different way. So either primary HLH or the secondary mass, they have different underlying mutations that they can drive, and the infection probably can trigger the IL-10. And in some kind of acute or in some EBV or CMV driven HLH, we think probably the IL-10, virus IL-10 plus the endogenous IL-10 probably really a kind of a major driver for another kind of subtype of HLH. As you know, interferon gamma now there are nice kind of therapies available. IL-18BP is another agent in the clinical trial. So we think probably the blocking IL-10 will be interesting kind of a new target for therapy in the HLH or mass disease. With that, I would really like to thank first the Histio Society for 2 grant support, keep us working on this side project in my lab, and we are leukemia lab actually. So Yuting, postdoc, and student Chen working on this project, and Ray and Yoshi did the HIFA alpha work back 4 or 5 years ago. And really, I would like to thank Dr. Mars, Pikasik, Mike Jordan, and Kumar for helpful discussion for this project. As you know, we have a Center of Excellence for the HLH research here. Transgenic Coral made the mouse, and also for the patient part, we collaborated with Jianfeng Zhou's lab in China, Wuhan, China, for the secondary HLH related profiling. Thank you for your attention.
Thank you very much. So now we have a couple of minutes left for questions. If anyone has a question. I actually have a question for Dina. And do we know before the grant process each year how many grants will be given? Like if it's going to be 5, 6, 7, 15, who knows?
We would love to have that number be 15, something to look forward to. Yeah, every year we would just determine the number of grants that we can based on the funding that we receive. So we typically have a sense of that going into the year. Historically, it's been somewhere between 3 and 6. And the more that we can fundraise, the more we can grow that program. And so that number to keep in mind for this year, as well as future years, somewhere between 3 and 6 is where we aim based on, and then of course we'd like to grow that as well.
Okay, thank you for the clarification. Do you have any more questions? Perhaps there's a question for Dr. Wang. Is it difficult to get samples from China? It's a question from.
Yeah, yes. So basically, because of they have a genetic, some material resource protection kind of law regulations. So yes. So basically, we use the same platform. That's some kind of profiling the basically it's a cytokine profile. So it's very hard to get samples, but they provide that's part of data. We are working mainly on the mouse modeling.
Thank you. So if there is no more questions left, just one quick check. I don't think so. So just to remind you that you can start shopping with Amazon Smile at our Amazon Smile link, and then you can donate at no cost to you. And then of course you can become a member for the Histiocyte Society, and your active involvement in the society will help us more quickly and better treatments to find a cure. Just there is one other question before I wrap up. It's still from Violet. Is HIF1A glycosylasis a possible diagnostic target? This is, I think, a question to Gang as well. Dr. Gang, do you see the question from Violet?
No. From OQA, okay. Hyperglycemia possible diagnostic target. Yes. Yes, I think so, because if you are familiar with the macrophage and the T cell in that kind of field, the activation of T cell macrophage, they can see from the serum, and even some kind of diagnosis tools like the LDHA level, because they can secrete it to the serum, can serve as a marker for the activation. So I think that's I don't know in the clinic, I'm not a kind of MD, whether they measure the LDHA, but in certain disease, definitely for the high glycolysis, they use LDHA to measure that. Could be a biomarker for that.
Okay, thank you. And so just please, everyone, try to complete the session survey, which help us improve the future virtual events like this, and it's important to have a feedback on the survey that will display after you exit this session. And I think that's about it. And thank you everyone for the very nice presentation and being there today, even if it's virtual. Thank you.
Thank you.
Bye-bye.